So I'm really happy today to be able to initiate some discussion about a topic that we've been thinking about for some time, but just lately managed to put in writing. So here we want to ask what we can learn from studies that look at sets of correlations between brain maps and what might be some limitations and potential solutions to these approaches. So this is a topic that goes beyond just histology and big brain, but still has some implications for it, given how it's been such a key and important resource in grounding other measures of brain organization into cytoarchitectonics. But before getting into that, I just wanted to dedicate this talk to the memory of Johnny Smallwood, who unfortunately passed away last week. So I'm very grateful to have worked with Johnny on this work, actually. He provides some really wonderful feedback in clarifying the different arguments that I'll be presenting today. So on behalf of myself and my co-authors, I just wanted to highlight how his presence will be really missed in the neuroimaging and cognitive neuroscience communities. So as neuroscientists, we want to obviously understand how the brain works. And to do that, we have to understand its different parts and how these different parts are related to each other. And one way to look at this is by constructing these brain maps that we see on the screen here, which will represent this anatomical distribution of values that reflect a given property of the brain. And we can see that these maps capture properties that range across different scales, different modalities, different states, different populations, and reveal these shared patterns of brain organization across individuals. And with the development of different tools like neuromaps and HIBALL maps, this has really facilitated how we can access, analyze, and visualize these different maps. And it's become increasingly easy to see how these different maps might be similar or related to each other. And the reason why we might want to do this, to see how these maps look alike, is because we can think that different properties that are expressed in an anatomically consistent way might share some common influences or common mechanisms. And this similarity will often be quantified, as we've seen in previous work, with simple correlations to assess the spatial correspondence that we see between two brain maps. So here we refer to this approach as data contextualization. And we can do this in different ways. So we can have a more kind of hypothesis-driven approach where we ask, okay, I know that these two properties of brain organization, for example, cytoarchitecture and myeloarchitecture, should be related to some extent, but really how similar are they? So we can attach kind of a magnitude to the similarity. And we might want to ask, how specific is this relationship? So is it really a stronger relationship that I would expect between two brain maps versus another map from a given set? But if we have access to kind of a broader set of different maps, we can also leverage this broad search space to see, or rather explore different candidate biological mechanisms for a feature that we'd like to decode. And the pipeline to achieve this often looks something like this. So we'll start with this source map, or a map that we'd like to better understand or decode, and first align it to a standard template space that's suitable for our analysis. So here I'm showing the first principal component of task activation patterns from neuroscience. And then to assess the similarity of our source map to a set of target maps, we'll systematically cross-correlate this source-target pairs on a region-specific basis so that we generate one correlation coefficient for every source-target pair. And we'll apply then some adapted statistical tests for this type of analysis, like spin permutation tests, to see if the correlation that we obtain between this source-target pair is statistically significant. And if we look at, in this particular case, the correlations that we obtain, so we see from this specific pattern of source-target pair correlations that these task co-activation patterns seem to co-localize with different microstructural territories. They seem to be recoverable in these task-free states, and we can also use it to dissociate these sensory areas from more association-type cortex. But a key question here remains is that what does it really mean that we found this specific pattern of correlations versus another pattern? And in this particular case, for example, we can ask if finding these correlations helps us to understand why or how these cognitive topographies are organized the way that they are in the brain. And it's difficult to say with this purely correlational approach because a lot of these brain maps that we have access to in these repositories tend to follow relatively similar patterns. So in the cortex, so on the left panel here, we see that these maps tend to cluster kind of in two general patterns. So we have this more kind of anterior-posterior pattern versus more unimodal to transmodal. And in the hippocampus, we see this same phenomenon to maybe a lesser extent. So if we look in the kind of upper left part of the scatter plot, we have a few maps that kind of cluster to follow this subfield organization of the hippocampus. So here the idea that we're kind of trying to communicate is that these more kind of exploratory analyses that make use of this broader search space often find some significant correlations with some other features of brain organization, because the same features tend to be kind of repeatedly expressed across different modalities or different scales. And if we translate this more to kind of a hypothesis-driven approach, we could also test specific hypotheses in kind of a more restricted search space of brain maps. So for example, here we could ask if two measures of cortical microstructure, for example, in vivo myelin-sensitive contrast like quantitative T1, are similar to postmortem histology. So for example, intensity sampled from BigBrain. And this could be used for validation, for example. And actually, we do find a significant correlation between our group map of quantitative T1 and a higher order feature derived from the BigBrain intensities. But we also see a comparatively strong correlation for a functional measure of brain organization. And the correlation that we obtain with each of these maps is statistically significant with the spin test. But again, we can ask, why is this the case? So it's a question that's difficult to answer with, again, purely correlational measures, because we can't really parse out the suspected causal links that are driving these shared spatial associations between the brain maps. But beyond this kind of more conceptual point, we think that there's also some technical limitations that might be making us find relationships between these different features that maybe aren't as strong as what the approach makes it seem. And a lot of these limitations have to do with how these brain maps are constructed in the first place. And these limitations might be kind of emphasizing certain patterns like this unimodal to transmodal differentiation that we see repeated across multiple maps. So in this interest of time, I'll just focus on a few of these limitations, starting with registration. So Jordan in his talk also touched upon a couple of these topics that I'll be talking about addressing this specific limitation, particularly looking at how the method that we use for registration might be more or less accurate across different regions that we're looking at. So as I showed, registration is really a key step in this data contextualization pipeline, since all of the maps that we're analyzing have to first be brought into a common template space. So obviously, if this step goes wrong, the validity of the maps that we're analyzing will be affected. So in the cortex, for example, we know that volumetric registration is maybe less precise than surface-based approaches because of the complexity of cortical folding, and this will affect how well regions in individual subjects line up in the selected template. But surfaces also have their own kind of set of limitations. So surface-based approaches will often use nearest neighbor interpolation when we're moving data from the high-resolution native mesh to a template space. And this will blur the source signals once we're kind of mapping them to the template. And the geometry of the template that we're using, for example, even with FSLR, will also, since it's not exactly the same across different regions, will make the magnitude of this blurring vary across different areas. So we can see how this blurring occurs in the FSLR space at the bottom figure here. And if we correlate these variations in blurring with maps from different modalities, we find that it's actually quite correlated with several features. Again. Okay. Also, there's some intrinsic features of cortical anatomy that might be overemphasizing some group-level patterns like the unimodal to transmodal gradient and boost some correlations that we see between these maps. So we know that cortical anatomy and folding is obviously not exactly the same across individuals, and this was highlighted in this really nice work that proposes this taxonomy of sulcal morphology. So here they found that these more linear sulci are more heritable, and they're typically located in more unimodal type of areas. These are the areas shown in purple. But more complex, less heritable sulci rather cluster in association cortex. And as we can see at the bottom, this pattern actually closely maps to the principal gradient of functional connectivity, so following this unimodal to transmodal axis. And this is important relating to the previous point on registration, since greater individual heterogeneity in these regions might make these areas more difficult to align across subjects. So this is maybe another point to consider for analyzing these brain maps. Oops. And Conversely, we also know that with some brain maps that are quite widely used, we have kind of the opposite problem where some data sets limit if it's even possible to quantify this inter-individual variability on a given brain property. So this is particularly true in data sets where we have rather small sample sizes, but even in larger samples from large neuroimaging data sets, we know that these data sets tend to overemphasize certain individuals with particular socio-demographic backgrounds, which challenges the generalizability of the inferences that we make from these brain maps. And there's always as well the issue of spatial autocorrelation when dealing with these brain map correlations. So as we know, brain maps tend to be autocorrelated, which means that areas that are closer together are more similar than areas that are further apart. And as a result of this, we're likely to find some significant correlations between brain maps just because they share this common smoothness. And different methods have been proposed to kind of correct for this. However, even though they're quite widely used, they still have some issues. So this was really nicely demonstrated in this recent work from Bretislav Misik's group, where the widely used spin test actually seems to distort the autocorrelation structure of brain maps. And these greater distortions tend to be associated with higher false positive rates. So we propose different solutions that we think might be able to tackle some of these issues and as well as others that weren't covered in this talk. And we do this from different angles. So we first propose that at the level of study design, it could be a good idea that we try to anticipate whether these type of brain map correlation approaches are best suited for hypothesis-driven versus more kind of exploratory type of analyses. And this can help guide downstream interpretations of the results that come from these analyses. And this could help avoid the use of ambiguous causal language that we often see applied in these type of approaches that would suggest that quantifying these spatial correlations between brain maps would reflect potentially shared mechanisms. And we also encourage the development and the adoption of frameworks that might be able to consider different aspects of the contextualization pipeline. Wrapping up. So this could include measures like registration accuracy, also how consistent these maps are across different data sets, across processing parameters or across individuals. And this could also be complemented by standardized metadata files that could be included in, for example, a JSON sidecar as we see with the BIDS architecture, so that features that went into the maps construction can be considered in our analyses as covariates, for instance. And we also encourage different analyses in data contextualization that might approach more kind of causal inquiry, or that can maybe consider simultaneously different aspects of between subject to within subject variability. So this could include hierarchical models, for example, that go beyond the implementation of just series of bivariate correlations. So this could also be complemented by the adoption of statistical techniques that I didn't have time to cover today, but that are able to kind of consider the heterogeneity of spatial autocorrelation that exists within brain maps. And lastly, it's easy to say that we need more data to kind of have broader, more diverse data sets, but often this is not really feasible or impractical in some contexts. For example, in data sets where there's really important technical challenges in building these resources. So maybe a potential solution, although this remains quite speculative, although we did see some really interesting work in this direction this morning, might be to exploit some advances in AI or deep learning to generate synthetic data sets that can help us augment the available data space for these types of analyses and maybe help build more complex models of these associations between different brain properties. So just to wrap up, we think there's a lot of exciting opportunities to address these limitations of data contextualization and get the most out of these approaches. And they've also delivered some very nice insights into principles of brain organization that are shared across individuals. But the goal is really more to think about ways that we can adapt these methods or complement them with other techniques to really help us clarify what's the nature of the interdependencies that we see between these different features or properties of brain organization, that these correlational techniques based on group average data extracted from different data sets might not be able to tap into. So with that, I'd like to thank all my co-authors, collaborators, and thank you for your attention, and happy to take questions.